This is just a short video to demonstrate the process of crossing over. So this process occurs during prophase 1, so only ever during meiosis 1. When prophase 2 takes place, no crossing over takes place. Because the premise of the process of crossing over is that it's when two homologous chromosomes, the homologous pair, come and exchange genetic material with each other. But if you think about it, during anaphase 1, the homologous pairs are separated. So if this is a homologous pair, one will go to one cell and one will go to the other cell, which means that they no longer are pairs in meiosis 2, so therefore you cannot have the process of crossing over. So this is how the process goes. You have your homologous pairs. Now we have 23 pairs, so this would happen in humans across all 23 pairs. So what happens is that these homologous pairs will come and lie next to one another, and they overlap at a point called the chiasma. So you can see these homologous chromosomes are lying next to each other, but what happens is these non-sister chromatids overlap at a point called the chiasma. So let me show you an example. So these are what you'd consider to be sister chromatids. So two blue chromatids are sister chromatids because they are identical to each other. Now, a red and a blue chromatid from opposite homologous chromosomes will be considered non-sister chromatids. So if I just point to a red and a blue one, that is a non-sister chromatid. So the chiasma is simply the point at which the chromosomes will touch each other. So that is, you'll have a chiasma at the top there and a chiasma at the bottom. This is the point where the chromatids will actually break off a portion and swap it with its homologous partner. So we call this the exchange of genetic information or genetic material between the two sister chromatids. So that would be the perfect definition for crossing over. Crossing over occurs during prophase 1. It's where homologous chromosomes lie next to each other and overlap at a point called the chiasma, and then genetic material is exchanged between the two homologous chromosomes. So when these homologous chromosomes separate, what happens is you end up seeing the homologous chromosomes each having some information from its homologous partner. So there's still one original chromatid on the paternal chromosome, but there's one chromatid that has some genes of its homologue. So the same thing happens on the maternal chromosome. You have an original chromatid and some with genes from its homologous partner. So that would be the last thing you'd have to state, that the homologous chromosomes separate so that each has one original chromatid and one chromatid with genetic material from its homologous partner. So again, crossing over occurs only during prophase 1, and it's when homologous chromosomes lie next to each other and overlap at a point called the chiasma. It might be in one or more places. In this case, it's in 2 places, and that leads to the exchange of genetic material between the two homologous chromosomes. This process introduces variation in the gametes because it allows the chromosomes to share genes so that all the sperm and egg cells that will be produced will be different from each other. It's the first process that introduces genetic variation during meiosis.